Welcome to This Explainer. Today, we're diving straight into an epic, microscopic battle that's been raging since the absolute dawn of life on Earth. On one side, we've got the meticulous, non-random force of adaptation. Think of it as a systematic sculptor of biological perfection. But on the other side, pure, chaotic, statistical luck. We're taking a hard look at the fundamental forces of population genetics. How do species actually change? Why do some really lethal genetic traits just stick around forever, while others completely vanish? And maybe most importantly, what actually happens when the sheer chaos of mathematics completely overpowers the survival of the fittest?
Okay, let's dive into this. Here is our structural roadmap for today. We'll start by decoding the gene pool, then move into the patterns of natural selection, followed by the chaos of genetic drift, and we'll wrap up with bottlenecks and founder effects.
Starting off with Section 1, Decoding the Gene Pool, the Evolutionary Reservoir.
To truly wrap our heads around evolution, we have to realize that it absolutely doesn't happen to isolated individuals. You literally cannot organically evolve during your own lifespan. But a population? A population definitely can. Think of the gene pool as a species' ultimate survival toolkit. It's the comprehensive repository of all their collective genes, and all the different variations of those genes, which we call alleles, housed within that specific group. An exceptionally diverse gene pool is exactly what gives a population the crucial adaptive potential it needs to survive things like catastrophic climate shifts or devastating diseases. And any quantitative mathematical change within this pool across generations, well, that isn't just a symptom of evolution. It's the direct, quantifiable measure of evolutionary transformation itself.
So, in population genetics, we track the frequency of specific alleles using variables, usually P and Q. Because a population is a finite group, P plus Q will fundamentally always equal 1.0, or 100%. It's a really elegant algebraic principle. If the environment shifts and suddenly makes allele P highly advantageous, that specific slice of the genetic pie is going to grow across generations. And obviously, if P grows, Q mathematically has to shrink. When we see these frequencies rapidly shifting, we are actively witnessing microevolution happening right in front of us.
Moving on to Section 2, Patterns of Natural Selection, the Force of Order.
So what's actually causing those genetic frequencies to shift? Well, natural selection doesn't just operate in one uniform way. Depending on local environmental pressures, how complex the habitat is, and what resources are available, selection can take one of 4 distinct patterns, and this brilliantly illustrates how the environment can sculpt a population's variance. Those patterns are directional, stabilizing, disruptive, and balancing. Let's walk through exactly how each one shapes the gene pool.
We're going to start with directional selection. This is when nature forcefully favors one extreme phenotype over all the others, basically pushing the population's average in a very specific direction. The absolute classic story here is the peppered moth during the Industrial Revolution. Before industrialization, light-colored moths were perfectly camouflaged against lichen-covered trees. Dark-colored moths were incredibly rare just because birds easily spotted and ate them. But then, coal factories coated all the trees in thick, dark industrial soot. Suddenly, the selective pressures completely inverted. The dark moths were now the ones camouflaged, and the light moths became the easy prey. As biologist Bernard Kettlewell rigorously demonstrated, that dark allele rapidly skyrocketed toward fixation, effectively shifting the entire population's phenotype to match their newly polluted environment.
Now contrast that with stabilizing selection. This is essentially nature's conservative force. It strictly operates in highly stable environments, and it actively punishes genetic extremes to maintain a perfectly adapted baseline. A really fascinating medical example of this is human birth weight. Historically, infants born with extremely low birth weights faced severe physiological challenges, while extremely large infants predictably presented highly dangerous clinical complications for the mother during birth. Because of this, natural selection heavily favored the intermediate baseline of about 3 to 4 kilograms, strictly minimizing variance to ensure optimal neonatal survival.
But what happens when the environment is complex and patchy? That's what triggers disruptive selection, which actively favors individuals at both extreme ends of the spectrum while selecting heavily against the intermediates. Take a look at the 3-spined stickleback fish up in the post-glacial lakes of Canada. You've got a robust benthic morph that is perfectly adapted for foraging in the shallows, and a slender limnetic morph adapted for filtering plankton out in the deep water. Now, if you're a biological hybrid, an intermediate form, you compete incredibly poorly in both zones. Basically, you're terrible at both jobs, so those intermediates effectively die out. Disruptive selection actively splits the population right down the middle, drastically increasing variance and actually paving the way for the creation of entirely new species. The fourth and final pattern is balancing selection. This one acts to preserve high genetic diversity by maintaining multiple alleles at a stable equilibrium. The most famous clinical example of this is the sickle cell trait in malaria-endemic regions. Here's how it works. If you have two normal alleles, you are highly vulnerable to lethal malaria. If you have two mutant sickle cell alleles, you suffer from severe sickle cell anemia. But if you're heterozygous, meaning you carry one of each, you gain this extraordinary life-saving partial resistance to malaria. Because this heterozygote advantage yields the highest overall survival rate, natural selection literally refuses to let either allele be lost. It maintains a stable, life-saving genetic equilibrium.
All right, moving to Section 3, the chaos of genetic drift, survival by pure chance.
Okay, let's totally shift our narrative away from these optimizing forces for a second. Because while natural selection systematically targets traits that improve survival, genetic drift is a force utterly, completely blind to biological utility. It is stochastic. It's purely non-directional. So the crucial point is that genetic drift is fundamentally driven by random statistical sampling errors during reproduction. It's wild to think about, but even if an allele is entirely neutral or even actively harmful to the organism, genetic drift has the exceptional capacity to arbitrarily increase its frequency just by sheer absolute dumb luck.
And I really need to warn you, over time, the relentless math of genetic drift inherently destroys genetic diversity. Because reproduction is ultimately a finite sampling of gametes, over thousands of generations, one allele will inevitably reach what we call fixation. Fixation is the absolute biological endpoint where an allele's frequency hits 1.0 or 100%. And when that happens, all alternative competing alleles are forced into permanent eradication. They just drop to zero. Drift steadily and relentlessly forces a population toward total genetic uniformity.
Which brings us to Section 4, bottlenecks and founder effects. This is when math completely takes over.
See, in massive populations, random statistical errors kind of just average themselves out. But in extremely small populations, this mathematical chaos completely overwhelms natural selection. First up is the founder effect. This violently alters allele frequencies the exact moment an exceptionally small, isolated splinter group leaves a massive mainland population to go start a new colony. Because these founders carry only a microscopic subset of the parent population's original genes, the new gene pool is instantly and profoundly skewed. Out of pure random chance, an allele that was exceedingly rare on the mainland can suddenly become artificially dominant in this isolated community.
Now, what's really interesting about this is looking at the human consequence of that math. The Old Order Amish of Pennsylvania were established by a remarkably small cohort of founders who practiced strict endogamy, basically cutting off gene flow from the outside world. Today, an astonishing 7% of this population is affected by or carries the allele for Ellis-Van Creveld syndrome. It's a severe disorder causing short-limbed dwarfism and heart defects. Globally, this allele's frequency is less than 0.1%. But geneticists traced it all the way back to a single ancestral immigrant couple, Mr. and Mrs. Samuel King, who arrived back in 1744. They were just asymptomatic carriers, but the blind math of the founder effect amplified their rare recessive allele to a massive 7%.
An equally catastrophic event is a population bottleneck. Now, this isn't migration. This is straight-up destruction. It happens when a catastrophic event rapidly decimates a massive population down to just a tiny fraction of survivors. Look at the northern elephant seal. In the 19th century, ruthless commercial hunting reduced the entire global population to an estimated 20 individuals clinging to survival off the coast of Mexico. Just 20. Today, they've numerically recovered to over 150,000, but they carry a permanent homozygous scar. Because they rebuilt from just 20 survivors, the entire massive contemporary population displays an absolute lack of genetic variation at numerous protein-encoding loci. That diversity is just permanently gone.
But perhaps the most terrifying real-world example of genetic drift's power is the modern African cheetah. Roughly 10,000 years ago, aligning right with the Pleistocene mass extinctions, the cheetah suffered an ancient, cataclysmic demographic collapse. The genetic bottleneck was so extraordinarily severe that today almost all modern cheetahs possess virtually identical major histocompatibility complex, or MHC, genes. And since the MHC is exclusively responsible for equipping the immune system to actually recognize pathogens, this absolute lack of variation leaves the entire global species incredibly vulnerable. Literally, a single virulent pathogen that bypasses one cheetah's immune defenses could theoretically bypass them all. It's a terrifying thought.
And that really forces us to look at the present day, doesn't it? When we step back and look at the actual mechanics of the gene pool, from the brilliant optimizing power of balancing and directional selection to the terrifying chaotic math of genetic drift, we have to realize that genetic diversity is absolutely a species' ultimate evolutionary armor. Which brings me to a final, provocative thought I want to leave you with. As modern human activity rapidly fragments habitats all over the globe, forcing countless species into artificial, microscopic bottlenecks, are we actively stripping away their evolutionary armor, abandoning their long-term survival entirely to the blind chaos of genetic drift? Thank you so much for joining me on this deep dive into population genetics. I hope you found it as fascinating as I do, and as always, keep learning.